Chapter 2, Sexual Reproduction in Flowering Plants. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us, are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants shows sexual reproduction. A look at the diversity of structures of the inflorescence, flowers and floral parts shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the process of sexual reproduction in flowering plants, that is angiosperms. 2.1 Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In class 11, you have read the various parts of a flower. Figure 2.1 will help you recall the parts of a typical flower. Can you name the two parts in a flower in which the two most important units of sexual reproduction develop? 2.2 Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a whorl of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. 2.2.1 Stamen, microsporangium and pollen grain. The two parts of a typical stamen, the long slender stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen, each from 10 flowers, that is, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of such stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed and each lobe having two theca, that is, they are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided, that is tetragonal, structure consisting of 4 microsporangia located at the corners, 2 in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed within pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by 4 wall layers, the epidermis, endothecium, middle layers and the tapetum. The outer 3 layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm, and generally have more than one nucleus. Can you think of how tapetal cells could become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell, that is PMC, through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad. As the anthers mature and dehydrate, the microspores deassociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grains represents the male gametophyte. If you touch the opened anthers of hibiscus or any other flowers, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometer in diameter. It has a prominent two-layered wall. The hard outer layer, called the exine, is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has prominent apertures called germ pores where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of a germ pore? The inner wall of the pollen grain is called the entine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by plasma membrane. When the pollen grain is mature, it contains 2 cells, the vegetative cell and the generative cell. The vegetative cell is bigger, has abundant food reserve and large irregular shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this 2-celled stage. In the remaining species, the generative cells divides mitotically to give rise to the 2 male gametes before pollen grains are shed, that is 3-celled stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and racehorses. Once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is a highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae, and Solanaceae, they maintain viability for months. You may have heard of storing semen, sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen minus 196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs. 2.2.2 The pistil, megasporangium, that is ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistil may be fused together, syncarpous, or may be free, apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated, slender part beneath the stigma. The basal bulged part of the pistil is the ovary. Inside the ovary is the ovarian cavity, that is the locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you studied in class 11. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be one, for example wheat, paddy, mango, or can be many, for example papaya, watermelon, orchids. The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Opposite the micropylar end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell MMC in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergo meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of 4 megaspores. Female gametophyte. In a majority of flowering plants, one of the megaspore is functional while the other 3 degenerate. Only the functional megaspore develops into the female gametophyte, that is the embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. What will be the ploidy of the cells of the nucellus, MMC, the functional megaspore and female gametophyte? Let us study formation of the embryo sac in a little more detail. The nucleus of the functional megaspore divides mitotically to form 2 nuclei which move to the opposite poles, forming the 2 nucleate embryo sac. 2 more sequential mitotic nuclear divisions result in the formation of the 4 nucleate and later the 8 nucleate stages of the embryo sac. It is of the interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not allowed immediately by cell wall formation. After the 8th nucleated stage, cell walls are let down, leading to the organization of the typical female gametophyte or embryo sac. 6 of the 8 nuclei are surrounded by cell walls and organized into cells. The remaining 2 nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. 3 cells are grouped together at the micropilar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The synergids have a special cellular thickenings at the micropilar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. 3 cells are at the chalazial end and are called the antipodals. The large central cell, as mentioned earlier, has 2 polar nuclei.